Start now. All right. Well, thank you, Dr. Procko, for coming back basically to USD and talking again to our students about your really, really interesting work. To get started, why don't you tell us a little bit about your background, and to make this personal, why don't you tell the folks, what you like about science, why you enjoy being a scientist, what kind of drives you as a scientist, before we get started with the details of the paper.
Okay. So, I mean, I was, like most little kids, I was interested in nature and my surroundings. I grew up by the beach, which many of your students probably have done so as well. So I was always fossicking down in rock pools, and from that just general interest of nature, it grew into, wanting to understand things at a molecular level. And then it was kind of just a natural progression. What I like about, I can tell you what I don't like about science. What I don't like about science is a lot of the politics and, getting work out into press, trying to get funding, it does wear you down a little bit. But, at the same time, I genuinely feel that what I do is important. I genuinely feel that I am contributing as best I can to important medical problems in the world. And so it's that real world impact which drives me.
Yeah. Excellent. Thank you. I appreciate that. So why don't we start with the big picture first? Where do you think the biggest impact, not just the results, what binds to what, but what do you think the biggest impact of your paper might be?
I don't know, because everything's moving so fast. I'm heavily focused on trying to make therapeutics, biologic therapeutics that will bind to receptor binding sites on the novel coronavirus to neutralize it. But I recognize that getting to that point in a clinic, there's a lot of hurdles along the way. That's going to require big players, not just me and my little lab, which is now mostly shut down to enforce social distancing, but it's going to require big players to step in. Where the more immediate impact may come is in the use of some of these high affinity reagents in diagnostics, also as controls in diagnostics, in what are known as ELISA assays. So there may be small steps along the way where it can contribute, because getting to the clinic, that's a challenge. In the best of times, it's a challenge.
Yeah, exactly. In terms of diagnostics, it would be kind of interesting to see a fusion receptor being used, whether it's to look for evidence that somebody's been affected or for the actual presence of this, right? Obviously, the former is usually an antibody. Do you think that this would be helpful, not just so much in point of care to find out when somebody is affected, but do you think this might be helpful in determining post-infection?
I don't think post-infection, other than as a control reagent on the, so even here at Illinois now, we're starting to develop, we will be developing what are known as ELISA assays to look for the presence of antibodies. Mostly, this is going to be together with the local hospital to see which healthcare workers have already been infected, maybe never even knew it, and mounted a good immune response, and so therefore are safer to return to work. So the reagents I have can be used just like antibodies. They still need some more validation with, for example, serum controls from people that post-infected people, but they can be used just like antibodies. But in terms of diagnostics while you are actively infected, then again, just like antibodies, they can be used for rapid tests to see are there viral antigens present in an infected individual.
What kind of platform besides an ELISA might that look like?
There are multiple platforms. The best would be something like a stick. If people are familiar with the kinds of paper sticks that are used for, say, pregnancy tests, of looking at hormone changes. You know, there are ways where you can have capillary action, where you draw up, you know, some sample onto you know, special filters, and if there's a reaction with your antibody or your antibody-like reagent, such as one of my engineered receptors, then you see some, you know, precipitate, something happens, and those tend to be rapid diagnostic tests where you get a result within, you know, tens of minutes. So that would be the ideal for a point of care diagnostic.
That would be great. All right, so let's talk a little bit about the paper. For a long time, we've known that whether it's the SARS, and then recently with the COVID-19, that the spike protein binds to ACE2. So how does your paper further our knowledge of that?
So the interaction affinity between the virus and the receptor in the original SARS virus, probably you're looking at many tens of nanomolar. So to put that into perspective, you know, it's moderate, but not very tight. This new coronavirus, what's known as SARS-2, it binds with higher affinity, and maybe that's important for it being more infectious. So binds with higher affinity, but again, depending on what numbers you look at, it's moderate. I wouldn't say super tight. So to have a really potent therapeutic, you want it to bind with very high affinity, in which case you probably need to do better than what we already have. And you also need it to be specific. I do not yet address specificity. I hope to address specificity, but don't yet. But what I can do is improve you know, binding signal. I can improve affinity. And what you don't yet see on the preprint, but students will see in maybe even the days ahead, because I put the data up very fast because I thought it was important for people to see it. But I've now gone and started combining mutations on a soluble form of the receptor. And it's clear that you can, you know, if you hit the right combinations, you can start to improve binding affinities by maybe multiple orders of magnitude. And that means that, you know, I don't measure exact affinities because the assays I'm using are more for speed. But based on the numbers that have been published, you know, we might now be looking at picomolar affinity, which is very tight. So wow. That's really where you want to be to have an excellent inhibitor. So I think we can get there, and then it will be addressing specificity, safety, and production.
Right. So to me, whether it's specific for SARS-1 or SARS-2 doesn't matter. It would be, as a therapeutic, would be block both of them.
Yes.
But specificity, knowing that you have a couple of amino acids extra in the RBD and a couple of other mutations, do you think you can hit it as a diagnostic so it would be able to tell the difference between the two? Or are those enough structural differences between SARS and SARS-2?
I don't yet know. So that I don't know the answer to. Ideally, you would actually want to hit both just as well. In fact, not only would you want to hit both SARS-1 and SARS-2, you would want to hit any future coronavirus that uses ACE2 as its receptor, so that way you are prepared for the next emergent zoonotic infection, the next spillover event that happens. And you need to keep in mind that, you know, in some ways, where we are is not unexpected. In the past 2 decades, less than the past 2 decades, there have been 2 major spillover events of coronaviruses into humans. One was the first SARS event, and then the second one is a related infection, uses a different receptor, but a similar receptor, called MERS. Mm-hmm. So if in 2 decades you see these 2 major spillover events, then, you know, it's not surprising that they're going to happen, and they're going to happen again. When I talk about specificity, the main thing that we need to know is that we don't bind other proteins in the human body. So my specificity needs to be for viruses and not for endogenous human targets, because then you will have off-target reactivity and side effects.
Very good. All right, let's get to some of the paper here. I'm going to share my screen so you can kind of your picture will be seen up here, but the main screen will show the picture here. So let me go ahead and share that. All right, so here's your preprint. Where did this actually get submitted to?
It hasn't been submitted yet. Okay. So my feeling was that that the data here, it's very nice data, but deep mutagenesis, the method that's used here, it can be noisy. So I wanted to feel confident before I send it off for publication that I'm not putting out there, false information.
Right.
So, but at the same time, I felt that the data was of sufficient public health importance that it needed to be there. So I put it out, and in the 2.5 weeks since this was posted, I've been working on validating mutations. I validated 30 single mutations, all of them as predicted by this experiment do indeed enhance binding between ACE2, the receptor, and protein S of the virus. So that was great. But what I also learned was that the effects were very small, just very disappointing, because this paper is not quantitative.
Right.
It's because something's enriched in a selection. I have no idea. Does that mean it's 2 times better, 3 times better? I have no idea. So the effects were very small. But at the same time, I then went and started combining mutations, which is now going to be very important. I'm doing it by intuition, just because I look at lots of structures, so I have a general feel of what I can combine without what's known as epistasis, where the mutations may have negative interactions when they're put in combination. So I've combined some, and that's where I can now see really nice improvements by orders of magnitude. So it's not yet submitted for publication. It will be. It will be soon.
Oh, that's exactly what the preprint kind of thing is for.
Yes, exactly.
And bravo for you to be willing to share this data so early, and especially in an era of getting scooped means an entire career.
Yes, I think in this area that's true normally, but in this area of research, I think everybody's paying attention to the preprints. So people see it, they recognize it. We did rush out very fast processing. We got it done in a few days, a patent on some of this. So, we do have some protection of intellectual property.
Okay, let's talk a little bit about, I'm showing the figure here. Can you just kind of describe the deep sequencing? You did a really nice job when you were talking about it at USD, but not all the students in my class or who might be seeing this later on were at that seminar. So could you talk a little bit about it and kind of walk us through the big mutational landscape that you have here?
So the idea is quite simple, and it's been developed within the past 10 years, these methods, where if you have a selection strategy, in vitro selection or a directed evolution experiment, where you are applying some pressure, some selection on a variety of mutants that are all together in a population. So you've got all of these mutants together. You select out the ones that have the activity you want. In my case, it's all fluorescence-based selections for binding, to look for the very best binders. You select those mutants out, and then you deep sequence what mutants you pull out. And you also deep sequence all of the mutations that were present in your population to begin with. So you can see, I had some mixture of, in my case, several thousand mutations. I can then see after my selection which of those mutations have increased in frequency, which mutations have decreased in frequency, and then you can do all the calculations. So in a single experiment, you can find out information, relative phenotypic information on thousands of mutations simultaneously. So typically what we see is that most mutations are going to be deleterious. That makes sense. That most proteins have evolved to be optimum for their activities. So you hit a protein with a mutation, usually it's not a good thing, easy to break something. What we're interested in are finding the usually rare mutations that make something better. And on this occasion, there's quite a few of those mutations because the receptor, the human receptor, never evolved to bind to the virus. So because it hasn't evolved to bind to the virus, it's not yet at an optimum sequence for that role, because that's not its purpose. Its purpose is to regulate blood pressure and blood volume. So therefore, I found lots of mutations that increase binding to the virus that we can take advantage of.
Very good. Can you explain on a basic level how you made all these mutations? Because many of our students will know how to do site-directed mutagenesis, but that's one at a time. You made how many mutations and how did you do it?
Yeah, so it's all PCR-based methods. The exact PCR-based approach that's used is called overlap extension PCR. It has similarities to a method that most students have used called quick change, but it tends to be a more robust method. And what we do is we incorporate degenerate codons. So at each position that we want to mutagenize, rather than encoding in the oligonucleotide primers used in the PCR, rather than encoding explicit nucleotides, A, C, T, or G, instead we encode in the primer N, 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 or something similar. So you now have a codon in your mutagenesis primer that has a mixture of all the different possible bases. So then you make a mixed PCR product. And then you do this at every position, which sounds like a lot of PCR reactions, but with a 96-well plate, it's really not. You can just set this up with a multi-channel pipette. It takes doing all the main delay in doing this is actually running the agarose gels to check all the products, but it can all be done in a few days to make the library. So here, this particular library, I targeted, if I recall correctly, I think it was 117 positions.
I think so.
And then by using degenerate codons at each amino acid position, I have all 20 possible amino acids available, as well as stop codons. So you have your wild type sequence, and then you have your 19 single amino acid substitutions plus a stop codon. So you would have 117 times 20 different possible variants that are being generated. And it's all a mixed population. They're not tested individually, mixed population, where you then do your selection to pull out the best stuff, and then you just deep sequence to see what goes up, what goes down.
Excellent. That's really amazing. And I'll be working with the students to look at the control experiments and how you selected for the positives and the negatives, or the things that we're binding. Could you kind of talk about the various preferences that you found that you highlighted in the paper, including some of the impact of glycosylation and what you think that glycosylation might be doing?
So the library was not random, because I can choose where I target primers towards. So the primers were deliberately targeted to the known interface. At the time, I should point out the structure of the SARS-2 receptor binding domain of the spike protein bound to the receptor ACE2 hadn't yet been solved. So I didn't have a structure, but you knew enough to know that it's going to bind similarly to the original SARS virus. So I used the known structures for SARS virus 1, and I decided to target mutagenesis towards the interface. So residues of ACE2, the receptor, that bind to the spike protein of SARS1. In addition, I also targeted the cleft of ACE2, where it interacts with angiotensin peptides. And the reason for that was it may be, this is debated, but it may be desirable to have an engineered receptor which doesn't interact with its normal endogenous substrates to thereby try to minimize toxicity and side effects. That's debatable because it may be that actually having enzymatically active ACE2 gives benefits in acute respiratory distress. But I decided to still also target the cleft to see what tolerance was allowed there. And in many ways, I'm glad that I did have a broader library because many of the mutations picked up to enhance binding to the spike protein are not at the interface. They're often a little distal, what we call second shell mutations. So they're like one layer removed. And even though I wasn't expecting mutations in these regions, they're mutations that you can look at the structure and you can rationalize, yes, this makes sense for stabilizing the conformation and the fold of the ACE2 protein. Because if your protein, ACE2, is not super well folded, let's say it breathes a little bit, so it's got some conformational dynamics and motion to it, then what that means is when you bind to a receptor, you now are forming a rigid complex, and that has an entropic penalty for locking in the conformation of ACE2. So if you lock in the conformation of ACE2, you pre-stabilize it by mutations, then now you can get better binding. You've got better presentation of the right conformation. So that's many of the mutations. That's most of the mutations. There are also some mutations at the interface. So we do find some specific mutations at the interface. And then the real hotspot for hitting was a glycosylation site. My gut feeling is that the sugars just probably get in the way. The sugars, so glycans, even though the structures only show, a couple sugars, that's simply because the structural biologists in the electron density, they only see good electron density for a couple sugar molecules. But there are many more sugars that are there. Glycans are big attachments on proteins. So you've got these huge, massive oligosaccharides, all these sugars that are on the surface, and they're kind of just floating around, they just drift with water molecules in solution, so they can just sterically get in the way. So we hit one of these glycosylation sites. I think you just have better access for the receptor to get in there and bind the target surface.
Okay. I'm going to stop sharing here. Just a second. I want to make sure I didn't miss anything. Yep, that was. So now it's just back to our screens. So that was a real tour de force. Just the idea of doing all those mutations and coming up with a high throughput way to look at this is just incredible. You talked a little bit about what you're doing now. What do you think you need to do to finish off this and then finally submit it?
I'll probably submit this weekend and also update the preprint on bioRxiv. The main things that I felt were absolutely essential was to validate single mutations to show that even in a format that's not a library, that's not all these mutations together, but you do targeted mutagenesis, test them individually. Yes, they really do improve binding. And then making some combinatorics, combination mutants to show. Hello, you still there?
Yep.
Oh, yep. Sorry, I just lost it on my screen. So doing some combination mutants to get a feel for how good can you make engineered ACE2 receptors. How much better can I make them? To really demonstrate to people that this can be a viable strategy. This can really work. So I can now show that you can probably make picomolar affinity binders, super tight binders. So I think that's enough to submit the paper. What I'm continuing on with, though, is I have made fusions of all of these engineered receptors to what are known as the FC regions of antibodies. This can give you immune effector functions. It can give you improved stability in serum, what's known as pharmacokinetics. It can also, in the case of one antibody class, immunoglobulin A, the immunoglobulin A class is the major set of antibodies that are secreted into the lungs during following lung infections. So it may also get you trafficking to the right side of the body for efficacy. So I'm producing all of these fusion proteins. I validated that they all work. I'm just now doing larger scale production for doing further characterization and really trying to push things to try to get to animal in vivo experiments as fast as I can to get this out there to meet the urgent need that's now there.
Well, then that actually brings up another question, which probably I shouldn't ask, but scale up and stability of the fusion receptor protein. Can you make a lot of this, and how stable is it?
So I don't know how stable it is yet. Can I make a lot? Within this kind of expression system, so this is not bacterial expression. This is made by mammalian cells in tissue culture. So you've got to have the appropriate expectations. I don't have exact numbers, but I have made various forms of the receptors fused to green fluorescent protein, in addition to fusions with antibodies and proteins of more relevance for a therapeutic. But I have these GFP fusions. And when you just look at the flask, by eye, the flask looks yellowish green.
Awesome.
And if you stick it on a blue light box used for looking at DNA and you look through a filter, you can see a ton of fluorescence. So there's clearly a lot of protein that's being made. That's a good sign, that you can probably make a fair bit, but it's going to need scale up. It's going to need larger scale production. There will need to be an assessment of manufacturability metrics, which means stability. So that's all needed, but it can all be done. And plus, I'm constantly depositing plasmids with a community depository called AdGene. So then those plasmids are available to any other lab in the scientific community to request them, to use them. I've also been sending out some plasmids already. So hopefully other people, if they see something promising, they can pick it up, they can start working with it.
All the best things that science is supposed to be. Thank you. When you talked, we talked a little bit before we started recording. You mentioned that you'll only run into 2 or 3 people in your building, in your lab building, because of the shutdown. So you just described a whole lot of work. How is that getting done?
I'm doing it. In the beginning, so I started this about a week into February. I think in the first week of February, I started this. the reason I started, so I do some work in my lab on viral glycoproteins and their receptors. And we've done work on HIV, on cytomegalovirus. And then I was looking for other viral targets late last year. And I thought, emergent zoonotic infections would be good viruses to go after. And I considered looking at SARS, but I thought, SARS came in 2003-ish. It then disappeared. Whereas there are other viruses known as Henipa viruses, on which the movie Contagion, if anyone's watched Contagion, it's based on a Henipa virus, something like a Hendra virus. So I thought I'd work on those. So that's, and I have a student in my lab working on that. And then suddenly this new coronavirus came out. And I was like, oh, I kind of wish now I'd been working on coronaviruses. And I sort of sat there, didn't do much. But then I spoke to a friend who was American, but was running a contract research organization in China. And he told me in this first week of February, obviously his company was no longer working. He'd been sent home. He'd managed to leave the country before all the lockdowns, so he wasn't stuck in China. And he told me how scared he was, and he didn't want to go back to China because he said that, the numbers that you're seeing out of China, they're not real because they've lost control of the situation in Wuhan. They really don't know what the real numbers are. And instead, look at not what the Chinese authorities say, but at what they do. And what did they do? They shut down regions of tens of millions of people very quickly, shut down all the transport, everything, shut down. And he said, when you look at that, and what the response that the Chinese Communist Party mounted, then this tells you what their concern level is. And that is frightening. And so after that conversation with him, I decided, I should start a project on coronaviruses. So I started ordering all the reagents to get the assays up and running. There were some delays there because everything was back ordered, because everybody was ordering the reagents. And then because all of my students were busy with other projects and because I am, the most experienced in my lab, I decided to begin things myself, do the experiments myself in the beginning. And then the idea was I could pass things off to lab members to really scale up after we got the initial data. I can no longer do that because I've had to send all of my lab members home because we need to enforce social distancing. So I'm now just doing it all myself in the lab. It's pretty lonely. It's not ideal, but at the same time, I also recognize from a public health perspective, I'm not sure I would want to be working around a whole lot of other people right now anyway. Yeah.
So one last question before I let you go. When did you start this project? Because a lot of this came out to light in December, January.
Yeah, so I only started in that first week of February. I'm
sorry, you started all of this. It is now for people who want to understand. What's the date today, Dr. Procko?
It is April 3. It is the 3rd of April.
It's the 3rd of April, and you started this
on probably around the 5th of February.
I just want people to understand how much work and how fast and how brilliant this has all been in such a short piece of time.
So the thing to keep in mind is that, if we were doing this 3-4 years ago, it would have taken us 6 months. But we have, the speciality of my lab is doing, in vitro selections in mammalian cells and deep mutagenesis. So we have the methods now really honed. They're really fine-tuned. And I had some help along the way, in particular the sequencing facility decided, when I told them, I gave them pre-warning that I have sequencing samples coming in. Sometimes it takes me a couple of weeks to get data back, but they cleared their deck. Everything else was put at the back of the queue. My samples were put at the front. So they really tried to help expedite things. Some of the companies, one company in particular that does gene synthesis for us, if you're working on COVID-19 related research, they made your samples their number one priority. So instead of waiting a couple of weeks again for reagents, it might be just a few days. And then, I really pushed through on it. I really worked hard to get it through. So it's doable, we can work fast. The main thing that actually limits how much I can do is that I have a wife who is a medical worker. She is a midwife at the local hospital. Already, she has had to deliver babies to a couple patients that were suspected of maybe having COVID-19. It puts a lot of stress in her work environment, as you can imagine, even though she's not in the emergency department. But still lots of stress, lots of extra work for her to go through as well. And then even though they've opened up the daycare for hospital workers, for essential workers, it's a bit of, we're a little afraid to send our children to the daycare. That's part of it. But on the other hand, our daughters also had a little bit of a respiratory infection, which if anybody who has a 3-year-old child is familiar, they have them all the time. So, we can't just take our children in at even the slightest sign of illness. We're not going to do that. We just won't take them in. So we're trying to manage both of our careers, both of our important work commitments while looking after children. I do a lot of work at nighttime. I do not get much sleep. So, yeah, it's tough. It's tough.
And
that's what you got to do.
You're not lucky, but it does kind of remind me, because it started with a conversation, your expertise, and then all this happened. It kind of reminds me of, I think it was Louis Pasteur who said, "Luck favors the prepared mind."
Yes, I know the quote. I don't know who came up, but yes, I know the quote, and that is true. That is absolutely true. That you need to be paying attention to what's going on, to recognize when you have an opportunity or when you have an important discovery. That is absolutely true.
All right, so I'm going to pause this now, but I want to thank you for all of your work and taking the time when, like you said, you've got children, you've got a wife doing important work, but to take time to talk to our students and then others, as you've already said, you'd be willing to share this. This is just fantastic. Thank you.
Okay, my pleasure.